Hello everyone, welcome to Pathomed. Today our topic is about Langerhans cell histiocytosis. These Langerhans cells are the immune cells. These are the dendritic cells or the antigen presenting cells present in the epidermis of the skin. The skin immune cells are called the Langerhans cell. So these are the dendrites, they give out dendrites. They act as antigen presenting cells of the skin. So whenever there's clonal proliferation of this Langerhans cell, it is a histiocytic proliferative disorder in which the Langerhans cell they proliferate abnormally and they migrate from this epidermis to any other systems, most commonly like bone or the lymph nodes. The genetics involved are most commonly seen mutations are BRAF mutations. BRAF V600E mutation seen in more than 50% of cases of LCH. Also, the other mutations can be MAPK pathways. Mitogen activated phosphate kinase pathways mutation can also be observed in few other cases of LCH. There are four forms of this Langerhans cell histiocytosis, the first being the Letterer-Sieve disease. Letterer-Sieve disease, it is a multifocal and multi-system disease. There are multiple organ systems are involved, and hence it is an aggressive form. It most commonly affects children below 2 years of age, and the most common sites being the bone and the skin. Whenever there is a bone involvement, for example, if skull bones or the facial bones are involved, there is concurrent CNS damage as well. The CNS damage can also be observed. There's neurodegenerative symptoms as well. The second form being the eosinophilic granuloma. As the name suggests, there is inflammatory infiltrate which is rich in eosinophils. Eosinophil cells are uh, like characteristically seen in this infiltrate inflammation. It is a unisystem disorder. That means it involves either one system. Most common being the calvarial defects. Calvarium means the skull. The skull bone is involved more commonly. Eosinophilic granuloma more common in the skull bones. The third form being the Hans-Koller-Christian disease. Hans-Koller-Christian disease, it presents as a triad. It has a triad of exophthalmos, eyes are involved, exophthalmos, and there is diabetes insipidus. Diabetes insipidus. So this means there's brain involvement. Brain involvement, pituitary gland is affected, and hence the ADH hormone is not released, for which that presents as diabetes insipidus. And thirdly, there's osteolytic bone lesions of the skull, osteolytic bone lesions of the skull. So it is a triad of exophthalmos, diabetes insipidus, and osteolytic skull bone defects. Hans-Koller Christian disease. The fourth form being the pulmonary LCH. The lung LCH is a rare disease. It most commonly it is associated with smoking, and this disease it regresses after stopping smoking. As soon as the patient stops smoking, this disease regresses. On radiographically, it is seen as cystic lung disease. These many cysts are present in the lung. Whenever these cysts rupture, the gas it escapes into the thorax and it may lead to spontaneous pneumothorax. Spontaneous pneumothorax. Coming to the microscopic picture, the Langerhans cell histiocytosis. So this side of the slide it shows you eosinophils. Eosinophils. This everything is eosinophils. So eosinophils can be identified as this with this orangish cytoplasm, pinkish orange cytoplasm. All this rich eosinophilic infiltrate. This all these cells are the eosinophils identified by their characteristic orangish cytoplasm. The other side, this everything, these are the Langerhans cells. A characteristic or interesting feature of this Langerhans cell is that its nuclear features. The nuclear membrane is not regular. It is folded. It is folded. Here it is folded. Does not have a regular contour. It is folded. Like this, it is folded. And few cells and few nuclei can also show this nuclear grooves. Nuclear grooves can also be seen in few of the cells. Few of the cells also show nuclear grooves. Mostly it shows nuclear folds. So Langerhans cells identified with their nuclear features like nuclear grooves and nuclear folds. Whenever there's a nuclear groove, it gives you an appearance of coffee bean nuclei appearance. This is characteristic of Langerhans cell histiocytosis. In this picture, the coffee bean nuclei appearance is very evident. It is very evident. Here you can see this nuclear folds as well. Coffee bean nuclei appearance are there. And coming to the electron microscopy, the very distinctive feature is Birbeck granules, presence of Birbeck granules, the tennis racket shaped granules in the cytoplasm of this Langerhans cells. This tennis racket, all these tennis racket granules present in the cytoplasm, tennis racket cytoplasm. These are the Birbeck granules. It is diagnostic of Langerhans cell histiocytosis. Don't confuse these granules with Birbeck cells. That is tennis racket cells, sorry. Tennis racket cells. Tennis racket cells are seen in embryonal rhabdomyosarcoma. These are the granules. Birbeck granules or tennis racket granules. Granules are seen in LCH. Coming to the immunohistochemistry, this LCH, it is positive for CD1A marker, membrane staining can be seen, membrane staining. Also, in few cases, multinuclear giant cells can also be seen. Multinuclear giant cell this is. And it shows Langerhans positivity. Langerhans is a CD207. CD207, also called Langerhans. It is a lectin protein. It is a lectin protein which is responsible for formation. This Langerhans is responsible for formation of Birbeck granules. So Langerhans is positive in LCH. Also S100 is also positive in LCH. These are the IHC markers. This is all about LCH. Thank you all.